We all know that our world runs on money, or what we call as currency. We do some job and expect money or currency in return, and sometimes we pay currency to get our jobs done. Similarly, in life sciences as well, all the living organisms are using a currency what is called as ATP. They are utilizing this ATP to carry out biological reactions, and this ATP is the central molecule in all biological reactions. Our body is producing almost 160 kg of ATPs per day. In today's class, we will be talking about these printing presses which are generating this currency. In today's class, we will be talking about the structure, function, origin, evolution of mitochondria and chloroplast. I am sure you have developed some basic understanding about this mitochondria and chloroplast in your lower standards. We all know that chloroplast is utilizing the energy of sunlight and produces ATP. These ATPs are utilized to synthesize carbohydrates like glucose, starch, and the animals are utilizing the energy which is stored in this glucose or carbohydrate to generate ATP. And this ATP is utilized by all plants and animals for synthesizing their biomolecules and carrying out their metabolic processes. Now let's dwell deep into the science of these organelles and study about them. We will not be going into the detailed biochemical aspect of the reactions which are involved in the ATP synthesis, but we will be more focusing about the structure and function of these organelles. In cell biology, a mitochondrion, which is plurally called as mitochondria, is a membrane enclosed organelle found in most eukaryotic cells. These organelles range from 0.5 micron to 1 micron in diameter. Mitochondria are sometimes described as cellular power plants because they generate most of the cell's supply of adenosine triphosphate or ATP. It is used as source of chemical energy in all biological reactions. Now let's see what a mitochondrion actually does. If we consider our cell as a big factory, various organelles have varied functions. For example, our nucleus provides blueprint for carrying out various jobs in the cell. At the same time, ribosomes are the production units that will synthesize protein for our cells. But all these biological processes require energy. This energy is provided by ATP. Production of this ATP is the job of mitochondria. Just like a furnace that burns coal and produces heat, our mitochondria burns glucose and produces ATP. This ATP is the energy currency of cells, and hence one can say that mitochondria empowers our cell by producing ATP, and that is why they are referred as powerhouse of the cell. In addition of producing cellular energy or production of ATP, our mitochondria are also involved in other processes. This includes cellular differentiation, cell death, apoptosis, the differentiation of the cells, cell cycle, and cell growth. Mitochondria have been implicated in several human diseases, including mitochondrial disorders and cardiac dysfunction. It also plays an important role in the aging process. Have you ever thought why this name mitochondrion was given to this organelle? Mitochondrion is actually a Greek word which is made up of two words, that is mitos and chondrion. Mitos means thread-like and chondrion means granules. This is because of the shape and appearance of this organelle under the microscope, and that is why this organelle is given the name mitochondria. Several characteristics make mitochondria unique. The number of mitochondria in a cell varies widely by organism and tissue type. Many cells have only a single mitochondrion, whereas others can contain several thousand mitochondria. The organelle is composed of compartments that carry out specialized functions. This compartment or regions include the outer membrane, the intermembrane space, the inner membrane, the cristae, and the matrix. Mitochondrial proteins vary depending on the tissue and the species. Although most of a cell's DNA is contained in the cell nucleus, the mitochondrion has its own independent genome. Further, mitochondrial DNA shows substantial similarity to bacterial genome. Apart from this, mitochondria always divide like binary fission in a bacterium. The first observations of intracellular structures that probably represent mitochondria was published in 1840s. The term mitochondria itself was coined by Carl Benda in 1898. However, Frederick Meves in 1904 made the first recorded observation of mitochondria in Nymphia plants. This was the first time when mitochondria was distinguished, identified, and we can say it was discovered. A mitochondria contains outer and inner membranes composed of phospholipid bilayers and proteins. The two membranes, however, have different properties. Because of this double membrane organization, the entire mitochondria is divided into 5 different regions. They include the outer mitochondrial membrane, the intermembrane space or the space between the outer and the inner membrane, the inner mitochondrial membrane, the cristae space which is formed by infoldings of the inner mitochondrial membrane, and the matrix or the space within the inner membrane. Now let's talk about the compartments of these organelles in an orderly fashion. First, we will be talking about the outer mitochondrial membrane. It contains large number of integral proteins called as porins. These porins form channels that allow molecules of 5000 Daltons or less molecular weight to freely diffuse from one side of the membrane to the other. Large proteins can enter the mitochondria via a large multisubunit protein called as translocase of the outer membrane. This translocase then actively moves them across the membrane. The intermembrane space. The intermembrane space is the space between the outer membrane and the inner membrane. Because the outer membrane is freely permeable to ions and glucose, the concentration of these ions and glucose is similar to that of the cytoplasm in the intermembrane space. However, as we have already seen that the large proteins are not freely permeable by the outer membrane, the concentration of these proteins in the cytoplasm and the intermembrane space is different. One of such protein which is localized in the intermembrane space is cytochrome C. The inner mitochondrial membrane. The inner mitochondrial membrane contains proteins with 5 types of functions. First, those that perform the redox reaction of oxidative phosphorylation. Second, ATP synthase which generates ATP in the matrix. Third, specific transport proteins that regulate metabolite passage into and out of the matrix. Fourth, the protein import machinery. 5. Mitochondrial fusion and fission proteins. The inner membrane is home to around 1/5 of the total proteins in the mitochondria. In addition, the inner membrane is rich in an unusual phospholipid, the cardiolipin. This phospholipid was originally discovered in cow hearts in 1942. It is usually characteristic of mitochondria and bacterial plasma membranes. Cardiolipin contains 4 fatty acids rather than 2, and that is why the inner membrane is highly impermeable compared to outer membrane. Unlike the outer membrane, the inner membrane does not contain porins, and that is why it is highly impermeable to all molecules. Almost all ions and the molecules require special membrane transporters to enter or exit the matrix. Proteins are ferried into the matrix via the translocase of the inner membrane complex or via OXA1. This OXA1 is also a type of transporter protein. In addition, there is a membrane potential across the inner membrane, which is formed by the action of the enzymes of the electron transport system. The inner mitochondrial membrane is compartmentalized into numerous cristae, which expands the surface area of the inner mitochondrial membrane, enhancing its ability to produce ATP. For a typical liver mitochondria, the area of the inner membrane is about 5 times as great as that of the outer membrane. This ratio is variable, and the mitochondria from cells that have great demand for ATP, such as muscle cells, contain even more cristae. These folds are studded with small round bodies known as F1 particles or oxysomes. Matrix. The matrix is the space enclosed by the inner membrane. It contains about 2/3 of the total proteins in a mitochondrion. The matrix is important in the production of ATP with the aid of the ATP synthase contained in the inner membrane. Matrix contains a highly concentrated mixture of various enzymes, ribosomes, tRNA, and the mitochondrial genome. It contains enzymes which are required for oxidation of pyruvate and other products which will synthesize the ATP. Mitochondria have their own genetic material and the machinery to manufacture their own RNAs and proteins. Functions of mitochondria. The most prominent roles of mitochondria are to produce the energy currency of the cell or ATP. That means it carries out the phosphorylation of ADP through respiration and to regulate the cellular metabolism. The central role of the mitochondria is the production of ATP. For this, it utilizes the Krebs cycle or what we call as citric acid cycle. Apart from the job of producing ATP, mitochondria is having other variety of roles. Energy production. The primary job of the mitochondria is the production of ATP. Mitochondria does this by oxidizing the various products of glycolysis, that is glucose, pyruvate or NADH. This process is also involving the oxygen, and that is why this entire process is called as aerobic respiration. However, in absence of oxygen, that pyruvate is converted to lactate or what we call as anaerobic respiration. However, the efficiency of the aerobic respiration is 13 times higher compared to anaerobic respiration. Pyruvate and the citric acid cycle. Each pyruvate molecule produced by glycolysis is actively transported across the inner mitochondrial membrane and into the matrix, where it is oxidized and combined with coenzyme A to form carbon dioxide, acetyl-CoA and NADH. This acetyl-CoA is the primary substrate which is utilized for the running of entire Krebs cycle or what we call as tricarboxylic acid cycle or the citric acid cycle. The enzymes of the citric acid cycle are located in the mitochondrial matrix, with the exception of succinate dehydrogenase, which is bound to inner mitochondrial membrane as part of complex 2. The citric acid cycle oxidizes the acetyl-CoA to carbon dioxide, and in this process it produces reduced cofactors, that are 3 molecules of NADH and 1 molecule of FADH2. These are the sources of electrons for the electron transport chain, and a molecule of GTP is formed. This GTP is readily converted to ATP and again recycled in the Krebs cycle. The redox energy from NADH and FADH is transferred to oxygen in several steps via the electron transport chain. This energy rich molecules NADH and FADH2 that are not only produced by the matrix but they are also produced in the cytoplasm by the process of glycolysis as well. Reducing equivalents from the cytoplasm can be imported into the matrix via the malate aspartate shuttle system of antiporter proteins or feed into the electron transport chain using a glycerol phosphate shuttle. Protein complexes in the inner membrane that are NADH dehydrogenase, cytochrome C reductase and the cytochrome C oxidase perform the transfer and the incremental release of energy that is used to pump the protons into the intermembrane space. This process is highly efficient and it ultimately reduces oxygen forming the water. However, during this process, some electrons might leach in and prematurely reduce the oxygen forming superoxide free radicals. These free radicals might damage the mitochondria and they are believed to be the probable reasons behind the aging process. Because of this electron transport system, hydrogen ions are continuously pumped in the intermembrane space. As the protein concentration increases in the intermembrane space, a strong electrochemical gradient is established across the inner membrane. These protons tend to return to the matrix through the ATP synthase complex and their potential energy is used to synthesize ATP from ADP and inorganic phosphate. This process is called as chemiosmosis and it was first described by Peter Mitchell, who was awarded with Nobel Prize in Chemistry in 1978 for his work. Heat production. We have already seen that electron transport system is continuously pumping protons from the matrix into the intermembrane space. This is leading to the generation of chemiosmotic potential. This is utilized by the F1 ATPase for the generation of ATP. However, during certain conditions, these protons are allowed to intentionally leak back into the matrix. This is leading to the generation of heat instead of production of ATP. This heat is utilized by the cells to fight the environment where cold climates are there. The process is mediated by a proton channel called as thermogenin or UCP1. This thermogenin is primarily found in the brown adipose tissues of our body, and as we all know, it is the brown adipose tissue which is responsible for generation of heat, which is in the case of non-shivering type. We know that our young ones or neonatals are not shivering, instead they are fighting to the cold with this brown adipose tissue, and this brown adipose tissue generates heat using this thermogenins. Origin of mitochondria. A mitochondria shares many similarities with the prokaryotic organisms such as bacteria, and that is why a hypothesis is proposed which states that this mitochondria might have evolved from some prokaryotic endosymbiont in the eukaryotic cells. A mitochondrion contains DNA which is organized as several copies of a single circular chromosome. This mitochondrial chromosome contains genes for redox proteins such as those of the respiratory chain. The circular structure is also found in prokaryotes, and the similarity is extended by the fact that mitochondrial DNA is organized with a variant genetic code similar to that of protobacteria. This suggests that their ancestor, the so-called protomitochondrion, was a member of proteobacteria. The ribosomes coded for by the mitochondrial DNA are similar to those from bacteria in size and structure. They closely resemble the bacterial 70S ribosome and not the 80S cytoplasmic ribosomes. This is because of the fact that the cytoplasmic ribosomes are synthesized by the nuclear DNA, and that is why they are of eukaryotic type, they are larger and heavier. Compared to this, mitochondrial ribosomes are synthesized by mitochondrial genome, which is similar to that of the bacteria, and that is why they are smaller, lighter and 70S type. The endosymbiont hypothesis. The endosymbiotic hypothesis suggests that mitochondria descended from bacteria that somehow survived endocytosis by another cell and became incorporated into the cytoplasm. The ability of this bacteria to conduct respiration in host cells that had relied on glycolysis and fermentation would have provided a considerable evolutionary advantage. Similarly, the host cells with symbiotic bacteria capable of photosynthesis would have led to evolution of chloroplast. The incorporation of symbionts would have increased the number of environments in which the cells could survive. This symbiotic relationship is believed to have evolved about 1.7 billion years ago. As we have seen that mitochondria is an organelle which is oxidizing the glucose and producing ATP, or we can say it is generating ATP. However, this glucose has to be formed somewhere. Formation of this glucose is done by the plants, and plants are having one specific organelle which is utilizing the energy of sunlight and forming ATPs. Further, it is using this ATPs to form the glucose, and this glucose is utilized by the animals, in fact almost by the mitochondria for the generation of ATP. So in a way one can say that the energy which is contained in the photons is utilized by the plants to produce ATP. These reactions happen in chloroplast. Further, these ATPs are utilized for the formation of glucose, and it is formed in the form of starch. This starch is then consumed by the animals. Hence, the energy which is present in the photons is first utilized by the plants by the organelle which is called as chloroplast. This is why they are called as autotrophs. Further, this energy is converted to glucose and other higher carbohydrates which are then consumed by the animals. These animals will then oxidize glucose by the mitochondrion, and then further they will generate ATP, and that is why they are called as heterotrophs. So this mitochondria and the chloroplast are the two organelles which are mainly responsible for generation of ATP. However, the chloroplast is using the energy of sunlight for the production of ATP, while the mitochondria is using the energy which is stored in glucose. In today's class, we have talked about our ATP producing organelle, the mitochondria. First, we have talked about the structure of mitochondria, then we talked about the function of mitochondria. We also talked a little about the evolutionary point of mitochondria. In the structure, we talked about the 5 compartments of the mitochondria, which are the outer membrane, the intermembrane space, the inner membrane, the cristae and the matrix. We have also discussed in detail about the functions of these 5 compartments. We have also talked about the Krebs cycle which is happening in the matrix. We have talked about the electron transport system and how it generates the chemiosmotic gradient. We have also talked about the F1 ATPase which generates the ATP. We also talked about the thermogenesis by the thermogenin which is using this chemiosmotic gradient for the generation of heat. Then in the later part of this class, we also talked about the evolution of this mitochondria, how a prokaryotic cell might have got trapped inside the eukaryotic cell and it had become the mitochondria.